All right, everybody, we're going to go ahead and get started. So before we begin, I just want to let you know that this session is being recorded, and if you wish to remain anonymous, please do not state your name, discuss your grades, or say any potentially identifying information. So as of today, all of you have taken your second exam. Congratulations. Whether or not you did well or bad, you still have a chance to make a really great grade on this course. All right, so today we're going to be covering enzyme regulation and mechanisms of allostery. This is stuff that you guys are going to cover next week. So there's that. And we're not going to do allostery and hemoglobin today, just because the lecture that you guys have on Thursday is going to be dependent on today's review. So if you can understand everything that we talk about today, next Thursday you guys are going to have a really great time in class. All right, so we're going to be covering enzyme regulation and mechanisms of allostery. And out of all the biochemistry concepts I've ever tutored, this is without a doubt the hardest one to explain. So if you guys have any questions, I want you to stop me. All right. And so before we get into that, these are some of the important questions that you need to think about when you're going over the slides on your own. And by the end of chapter 15, you need to be able to answer them on your own. So let's start with a really easy one. What do enzymes do? What do enzymes do? They catalyze reactions. What are enzymes made out of?
Proteins.
They're made out of proteins. What are proteins made out of?
Amino acids.
Amino acids. Okay. So when an enzyme or a protein binds a substrate, what happens to the enzyme structurally? Do you guys remember talking about the induced fit model? So what happens to the whole protein when it binds a substrate? A conformational change. I want you guys to remember that key term. Conformational change. All right, and so here we have this enzyme right here. It's called Hexo Kinase. What happens to this enzyme as soon as it grabs substrate, as soon as it grabs glucose? A conformational change. And if a conformational change is occurring, what do you think is happening to the interactions between the R groups in the enzyme?
Changing.
They are also changing. That's a really important fact that you need to remember. So let's go ahead and zoom in here. And let's imagine that right here, we used to have a lysine residue. Let's imagine that we had a lysine residue and we also had an aspartic acid residue right there. When this enzyme grabs glucose, we can almost say that they stop interacting. Just as an example of a conformational change. So they used to be interacting here because they used to be a lot closer, but now due to the conformational change, they stop interacting. Does that make sense? Are there any questions about that? Okay. So essentially we've broken a salt bridge. That's another key term. We've broken a salt bridge. All right, so how can we control the activity of the enzymes? In Unit 2, we learned about molecules which could slow down the performance of enzymes. What are they called? What are they called? Inhibitors. Good job. So all the enzymes that we talk about now, they can be inhibited, but they can be inhibited through different mechanisms, such as allosteric regulation. All right, and so this is the overview of mechanisms of regulation. What type of graph is this? It's a hyperbolic graph. What type of enzymes display hyperbolic kinetics? Michaelis-Menten enzymes. We are no longer going to be talking about Michaelis-Menten enzymes. Now we're going to be talking about enzymes whose graphs look like this. And this is what again? These are allosteric enzymes. All right, and what we just talked about, conformational changes, this is the big thing that you guys got to know about allosteric enzymes. It's the key reason why allosteric enzymes behave the way they do. All right, so how else can we control the activity of enzymes? This is what the entire chapter is about. The first way we can control them is abundance. What do I mean by abundance? Abundance of enzymes. What does that mean? Okay, the amount. So this is the amount. How can we control the amount of enzymes that are in a cell at a given time? There's a little image right here that can help you answer that. Through the transcription and translation process, right? So through genetics. However, that's a topic for genetics, the actual course. So for now, we don't really care about that. But this is just one way that we can control the amount of enzymatic activity, the amount of enzymes. If we have a lot of enzymes, what can we say about our Vmax? Is it going to be high or low? Higher. So if we have a lower amount of enzymes, what's going to happen to our Vmax? It's going to go lower. So controlling the abundance of enzymes is a way to control the activity. Okay. So the way that we can control the activity of enzymes directly, we can do it through covalent modification. And if you see here, covalent modification of an amino acid chain can activate or inactivate an enzyme. That's a really important thing for you to know. Just because something has been phosphorylated doesn't mean that it's been activated or deactivated. Are there any questions about that?
Can you repeat that?
Sure. So if we look at this image over here, let's look at this enzyme with this hydroxyl group. And this enzyme right here with this hydroxyl group, it's the catalytically active enzyme. Look at what's going to happen to it. This enzyme with that hydroxyl group is going to be phosphorylated. And now it is the catalytically inactive enzyme. Does that make sense? So we used to have an enzyme with a free hydroxyl group, and then if you look over here, it was phosphorylated. So it received a phosphate group, and now this enzyme is inactive. So does this mean that if you phosphorylate an enzyme, or if you phosphorylate an amino acid on an enzyme, does that mean that you inactivate it? Yes or no? For this enzyme, it does. But it's not true for all the enzymes. Does that make sense? Are there any questions about that? If anybody wants me to repeat that, just raise your hand. Do you have a question?
I'm assuming we're going to talk about which enzymes do that and which ones don't.
Later on in the course we will.
Okay.
But just as a general rule of thumb, if you have an enzyme, so let's say there's our enzyme right here. There's our enzyme, and let's say it has a hydroxyl group right there. Another enzyme, so a protein kinase, will take ATP, take one of those phosphate groups off of ATP. When you take a phosphate group off of ATP, what does it become? ADP. And this enzyme takes one of those phosphate groups and it's going to give it to this enzyme right here. What just happened to this enzyme? What just happened to this enzyme? It was covalently modified. What amino acid residue was covalently modified on this enzyme? Serine. What other amino acids can be phosphorylated? Tyrosine, threonine. So tyrosine can also be phosphorylated, and threonine, and also histidine, but your textbook doesn't talk about that, so let's not get into that. Okay, so if you phosphorylate an enzyme, does that mean that you've activated or deactivated the enzyme? It means neither. It depends on the enzyme. Good job. So does it depend on the enzyme, yes or no?
Yes.
Yes. So if you phosphorylate an enzyme, do you know if you've activated or deactivated it? It depends on the enzyme, so no is the answer. All right, so that's a really important point. What type of enzymes phosphorylate other enzymes? Kinases. So you see this protein kinase right here. This protein kinase takes this ATP, turns it into ADP, and what happened to that other phosphate group? So, yes, it gets attached to what? To the other enzyme. So do you guys see that? We have one enzyme, we have an enzyme, and what is it doing to another enzyme? It's acting on it. Are there any questions about that? This is a really important concept that you guys need to understand. Enzymes can act on other enzymes. Are we okay with that? Are there any questions about that? All right.
So is covalent modification only phosphorylation, or is there more?
There's more. Yeah, you can also transfer like acetyl groups and methyl groups, but the main one that you guys are gonna see is phosphorylation. Okay, and how do we remove this phosphate group?
An enzyme?
What type of enzyme removes phosphate groups? It's perfect, phosphatases. So kinases add phosphate groups, and what do phosphatases do? They remove them. And they remove them using what? Water. So they use water as a substrate. So whenever you're looking at this on your PowerPoint slides, I want you to look at the enzyme with the hydroxyl. I want you to see that if you go towards the right, like the right arrow is pointing, it's showing that it's getting phosphorylated. And if you remove that phosphate group, it becomes the catalytically active form again. But if you phosphorylate an enzyme, do you know if it's been activated or deactivated? It depends on the enzyme. All right, so one way that we can control the activity of enzymes is through covalent modification. Are there any questions about that? Yes.
So would phosphatase be lyase or transferase?
So a phosphatase, since it's using water to cleave, what do you think it is? A hydrolase.
Oh,
All right, so if an enzyme is covalently modified, does this mean that the enzyme has been activated or deactivated? What's the answer?
Depends.
That depends. All right, another form of covalent modification, this is the proteolytic cleavage of Zymogens. What are Zymogens? You guys haven't covered in the class, so I guess you shouldn't know. Okay, Zymogens are enzymes, but they're enzymes that are not active yet. So another way you can think about it is it's a baby enzyme. It's a baby enzyme that's just been born and it has no catalytic activity yet. Are there any questions about that? It could also be a protein. All right, so if we look here, we can control the activity of enzymes by synthesizing inactive precursors, which are called Zymogens or proenzymes. And Zymogens are basically inactive enzymes. What do enzymes do?
Catalyze reactions.
Enzymes catalyze reactions. Can Zymogens catalyze reactions? For the most part, no. All right, so activation requires specific proteolytic cleavage. What is proteolytic cleavage? What is proteolytic cleavage? What do you guys think that is? Cleavage of a protein. So to activate this, what do we need to do to it? We need to cleave it. So activation of Zymogens, which are baby enzymes, require proteolytic cleavage of one or more peptide bonds. And unlike other mechanisms, this is irreversible. All right, so I want you to look at this right here. Look at this picture with pro insulin and insulin. Pro insulin, anytime you see pro, I want you to know that that is a zymogen. So if you ever see pro, that is a zymogen. And is pro insulin active, yes or no?
No,
Why not?
because it's a zymogen.
Because it's a zymogen. Okay. So here's pro insulin, and this right here to the right of it is insulin, the active form. What do you see as the difference between pro insulin and insulin? It's missing what? It's missing the connecting peptide, also known as your C peptide. So insulin is missing the C peptide. When you guys get into your problem sets next week, do you guys have class next week?
Yes.
Okay. Oh, and then we have spring break, right? Okay. So whenever you see C peptide, I don't want you to get freaked out. That is just the part of the proenzyme that needs to what? That needs to be cleaved in order for what to happen?
Activation.
In order for activation to occur. So this is proinsulin and this is insulin. What is the difference between proinsulin and insulin? What is the difference between proinsulin and insulin? The C peptide has been cleaved. And is insulin active? Yes. Are there any questions about that? Any questions at all? Okay, so the hormone insulin is made as an inactive 86 amino acid precursor, proinsulin. So if you ever see pro, what do you know it is? Zymogen. Okay. You guys also talked about an enzyme called chymotrypsin. Chymotrypsin. Chymotrypsin has a zymogen version of it, and that is chymotrypsinogen. Alright, so if you ever see ogen at the end, that is also a proenzyme. Alright, so proteolytic cleavage of enzymes of the digestive tract are made as inactive zymogens. So the zymogen of chymotrypsin is chymotrypsinogen. Is chymotrypsinogen a zymogen, yes or no?
Yes.
Good job. So how do you know that it's a zymogen?
The gene at the end.
The gen at the end. So which is activated first by trypsin. So look at this. We have chymotrypsinogen, and then we have trypsin. And what is trypsin doing to chymotrypsinogen? It's cleaving it, and by cleaving it, it's doing what? It's activating it. So we have an enzyme acting on another enzyme. Does that make sense?
Yes.
Okay. So here we have chymotrypsinogen, which is the inactive zymogen, and cleavage by trypsin is turning it into pi-chymotrypsin, which is now active. But before it can become, I guess, even more active, what must first occur, going from pi-chymotrypsin to alpha-chymotrypsin? Self-digestion. So it's a little bit like cannibalism, but in a good way. Are there any questions? So in order for chymotrypsinogen to be turned into chymotrypsin, what must happen? Cleavage by trypsin. And then once chymotrypsinogen has been turned into chymotrypsin, what does chymotrypsin do to itself?
Digest.
Self-digestion.
So does that mean that like amino acid parts are going to come out of the
So thank you for saying that. So what this means is that the enzyme itself is going to cleave this, 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 and this. So it's going to cleave here and here. And it's going to lose those 4 amino acids. And now what we have is this active enzyme. And just out of curiosity, what are these gold or yellow lines right here? What do those represent? disulfide bonds. Perfect. These are disulfide bonds. And what type of amino acids make up disulfide bonds? Cysteine. And are disulfide bonds part of the tertiary or are they part of the primary structure?
Primary.
Part of the primary structure. You guys are on it. Good job. Okay, so far we've discussed, I think, two ways to, or one way to control the activity of enzymes. What's the first way that we discussed? Covalent modification. What's one form of covalent modification?
Protein.
Phosphorylation by what type of enzymes? Protein kinases. Okay, now we're talking about a different type of modification to the enzyme, covalent modification to the enzyme. Now we're talking about proteolytic cleavage of the enzyme. So do you guys see how this is changing the way that enzymes behave? By phosphorylating it, you can do what to an enzyme?
Inactivated.
You can inactivate it or deactivate it. And by proteolytically cleaving a zymogen, what are you doing to that enzyme? You can activate it. So this is what we're discussing. We're discussing the ways in which we can control the activity of enzymes, which is what we're supposed to be doing, right? The enzyme regulation and mechanisms of allostery. All right. Are there any questions about anything that we've discussed so far? All right, we're about to get into the tricky stuff. But before that, let's go into a little something sweeter. Enzyme activity can be regulated by other proteins. Do we agree with that? What does that mean? What does it mean when we say that enzyme activity can be regulated by other proteins? It can mean that. So it can mean that it's being regulated by substrate. But what this really says is that you can control the activity of one enzyme by another enzyme. Does that make sense? Are there any questions about that? So look at what we have here. So this right here is cyclic AMP dependent protein kinase. So cyclic AMP dependent protein kinase. So I'm going to put PK. What do you think this protein kinase, so this right here is a kinase. What do you think this protein kinase requires in order for it to be catalytically active? If it's a cyclic AMP dependent protein kinase. So it requires cyclic AMP. So look at this. This enzyme right here, is it active or inactive? The enzyme to the left. It's inactive, and check this out. These are subunits. These are regulatory subunits. When this enzyme has its regulatory subunits, it is inactive. Does that make sense? Yes.
So does the regulatory subunits cover the active site?
It could cover the active site, or it could shape the catalytic subunits in a way that doesn't let them react. Does that make sense? Okay. So these regulatory subunits don't necessarily have to cover the active site. They can just change the shape of the catalytic subunits so that they don't work. All right. So check this out. These regulatory subunits are going to have cyclic AMP covalently attached to them. And once cyclic AMP is covalently attached to them, what have we just done? So we've kicked off the regulatory subunits, and that's important. Once we've kicked off those regulatory subunits, what have we now made?
Active.
The active form of the enzyme. We have two catalytic subunits that are good to go. So when we say that enzyme activity can be regulated by other proteins, we mean that there's an enzyme which attaches cyclic AMP to where on this protein right here. So we have an enzyme that attaches cyclic AMP to the regulatory subunits. Once we attach cyclic AMP to those regulatory subunits, what happens to those regulatory subunits? They kick off, they dissociate, they detach, and what have we just created? Catalytically active protein kinase.
So is the CAMP also kicked out too?
It's kicked off with the regulatory subunits, yeah, but it's covalently attached. So when we say that the enzyme activity can be regulated by other proteins, we're talking about a protein which is taking cyclic AMP, more specifically 4 of them. It's taking 4 cyclic AMP, attaching them here, 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 and here, kicking off the cyclic AMP dependent protein kinase regulatory subunits, and making 2 active catalytic subunits. What does a protein kinase do? Phosphorylate using what?
ATP.
ATP. What type of enzyme is a kinase? Transferases. All kinases are transferases, but not all transferases are kinases. All right. So B, enzyme activity can also be controlled through allosteric regulation. So far, all we've talked about is how we can control the activity of enzymes through covalent modifications. What's the first covalent modification that we talked about? Phosphorylation. What's the second one? What's the second type of covalent modification?
Zymogens.
The proteolytic cleavage of zymogens. And once we proteolytically cleave zymogens, what happens to them? They are activated. Good job. All right. So now that we've discussed that, we can go into the hard stuff. Enzyme activity can also be controlled through allosteric regulation. This is key. The majority of this chapter is about the mechanisms of allosteric regulation. So if you guys are falling asleep, now's the time to wake up and pay attention. All right. So allosteric regulation is conducted by allosteric effectors. Allosteric effectors do what to allosteric enzymes? They can induce conformational changes, but even more simply and more generally, allosteric effectors are the things that act on allosteric enzymes. And there's 2 main types of allosteric effectors. There's allosteric activators and allosteric inhibitors. What is an allosteric site? It's a site other than the active site. So are these allosteric inhibitors and allosteric activators competing for the active site? Yes or no?
No.
No. All right. So before we get into that, I only want you to look at the left side of this, which represents glycolysis. Raise your hand if you remember glycolysis. All right. Well, don't worry. We're going to go through it plenty of times. So by the time that you go into your 3rd exam, you'll be very well versed in all of this stuff. Okay. So there's 2 main types of enzymes that we've discussed, but we haven't gone fully in depth. We've discussed allosteric enzymes. And what's the other type of enzyme that you guys already discussed many times? Michaelis-Menten enzymes. All right, so your allosteric enzymes are going to be mostly your regulatory enzymes. Your allosteric enzymes are going to be mostly your regulatory enzymes. What are the regulatory enzymes in glycolysis? You don't have to know their names, you can just tell me the numbers. Is it enzyme 1, 5, 20? The first regulatory enzyme in glycolysis is hexokinase. This is not an arbitrary fact that I'm telling you. This is something that you're going to want to know. The first regulatory enzyme in glycolysis is hexokinase, or glucokinase if you're talking about the liver. This is also something that you need to know. So glucokinase is in the liver. That's not what I wanted. All right, glucokinase is in the liver, and hexokinase is in pretty much every other cell in your body. Every other cell. All right, so is hexokinase a regulatory enzyme, yes or no? Yes. So what type of kinetics is it going to exhibit? Allosteric kinetics. So its graph is going to be, is it going to be a hyperbola or is it going to be sigmoidal?
Sigmoidal.
Sigmoidal, perfect. All right, the second regulatory enzyme in glycolysis is what? What is it? What is it? Phosphofructokinase, good job. So that's PFK1. This is another regulatory enzyme, and regulatory enzymes display what type of kinetics? Allosteric.
Is that the second one, or is it just?
Oh, what do you mean?
Like isomerase, is that one or no? Like
Oh.
one in between?
No, so the second one is not a, it's not a regulatory enzyme. Yeah. All right, and so the third regulatory enzyme in glycolysis is pyruvate kinase. And hexokinase, PFK1, and pyruvate kinase, they all exhibit allosteric regulation. They are all regulatory enzymes. And all these other ones, like phosphohexoisomerase, triose phosphate isomerase, aldolase, all the other ones that I didn't highlight, are those regulatory enzymes, yes or no?
No.
No, they are not. So are they going to exhibit sigmoidal curves?
No.
No, they are not. They're most likely going to exhibit Michaelis-Menten hyperbolic curves. So that would be these right here. Okay, so this right here is an example of what the curves look like. And regulatory enzymes have sigmoidal curves. All right, so this right here, this graph that you see on the left, this is the graph for phosphofructokinase 1. The 1 is important. So this is the activity of PFK1, and look at this. This right here, this blue line that you see on the screen, this is what the graph looks like when there's 0 micromolar of fructose 2,6 bisphosphate. All right, so here's PFK1, and PFK1 takes fructose 6-phosphate, it takes fructose 6-phosphate and it converts it into what? Do you guys remember? It turns it into, close, fructose 1,6 bisphosphate. All right, so all your glycolytic steps, you're gonna wanna remember those. So phosphofructokinase 1, it takes fructose 6-phosphate and it turns it into fructose 1,6 bisphosphate. And this right here, this blue line, represents what the graph looks like when there's just the enzyme and the substrate. So when there's the enzyme plus the substrate forming enzyme-substrate complex, you get enzyme plus product. But look at this. Look at this green line right here. And this is exactly what I mean by allosteric effectors. Look at this green line. Whenever you have 0.1 micromolar of fructose 2,6 bisphosphate, what has happened to the relative velocity? Has it increased or decreased?
Increase.
It's increased. So let's do this. Let's analyze this graph a little closer, because this is really important. Whenever we have 1 micromolar of fructose 6-phosphate, and remember fructose 6-phosphate is the substrate, whenever we have 1 micromolar of fructose 6-phosphate, we can say this is the velocity right here. We can say it's 20 micromolar per second. Does that make sense? Are there any questions about that? whenever we have 1 micromolar of fructose 6-phosphate, this reaction is occurring, and it's occurring at this rate, 20 micromolar per second. Are we okay with that? Are there any questions about that? Any questions at all? Whenever we introduce 0.1 micromolar of fructose 2,6-bisphosphate, so fructose 2,6-bisphosphate, what has happened to the velocity of the reaction? Let's use green. So whenever we introduce 0.1 micromolar of that, look at the velocity. It just shot up to about 83 micromolar per second. So what do you think this fructose 2,6-bisphosphate is doing to this enzyme? Is it making it work faster or is it making it work slower? It's making it work faster, and that right there is an allosteric activator. Good job. So for now, let's not care about what this fructose 2,6-bisphosphate is exactly doing on this enzyme. Just understand that this fructose 2,6-bisphosphate has made this enzyme do what? It's activated it. It's made this reaction happen a lot faster. So now this is super fast.
So are we saying in the blue line it's inactivated?
So the blue line represents when it's just the enzyme and the substrate. There's nothing in the mixture. But as soon as we add just a little bit of this fructose 2,6-bisphosphate, what has happened to the catalytic activity, the performance of this enzyme? It has increased. Okay, so that's one thing that I want you to note. Allosteric activators, they do what to regulatory enzymes? They what? They increase the catalytic activity. And do you guys see how this blue line right here is kind of S-shaped in the beginning? It's got a sigmoidal aspect to it. And do you guys see how once we introduce this fructose 2,6-bisphosphate, this the graph looks a little more hyperbolic, a little more Michaelis-Menten?
Yes.
So here's what I want you to note, and this is like the most important thing. As soon as we introduce an activator, this sigmoidal curve, it's due to cooperativity. We'll go into that in just a little bit. But this sigmoidal curve is due to cooperativity. That is so important that for those of you that are watching this online, and for those of you here, I really want you to remember this and ingrain this into your brains. Sigmoidal curves are due to what?
Cooperativity.
Due to cooperativity. All right, we'll go into cooperativity in just a little bit, but sigmoidal curves are due to cooperativity. Once you introduce an allosteric activator, what has just happened to the cooperativity? Has it increased or has it decreased? I realize that may be a little bit of a difficult question. What was that?
Decreased?
Did you say decreased or increased?
Decreased.
Decreased? I can't hear you.
Yeah.
Decreased.
Excellent work. Good job. So whenever you introduce an allosteric activator, you have just removed the cooperativity. You have just reduced the cooperativity. And now the enzyme behaves more like what? Like a Michaelis-Menten enzyme. So allosteric activators, they reduce cooperativity. Good job. All right, and so what happens when we increase that amount of fructose 2,6-bisphosphate? What has happened to the cooperativity then? What do you guys think? It's decreased even further. And so that's why the velocity increases. Are there any questions about that? All right, so we've just talked about an allosteric activator. And what is the allosteric activator of phosphofructokinase? What is the allosteric activator of phosphofructokinase? F26 what? Bisphosphate. Good job. So this right here is an allosteric activator. This is important. All right. So we've just talked about an allosteric activator, and now we're going to talk about an allosteric inhibitor. Okay, so let's look at the blue line again. This right here represents the velocity of PFK1. What is PFK1? What enzyme is that? Phosphofructokinase. So PFK1 is phosphofructokinase. And look at the velocity of this enzyme whenever you have 0.1 micromolar of ATP, or 0.5. So the relative velocity, we can say it's about 60 micromolar per second. When you increase the amount of ATP, what is happening to the velocity? So don't look at the green or yellow lines. I only want you to look at the blue line. When you increase the amount of ATP, what is happening to the blue line? It's decreasing. So what do you think ATP is doing to phosphofructokinase 1? It's an allosteric inhibitor. So therefore, as the concentration of ATP increases, the activity of PFK1 decreases. Does that make sense? If there's a little confusion, I can see why.
Is PFK1 the allosteric activator?
PFK1 is what? If it has ACE at the end of it, what is it? It's an enzyme. So PFK1 is an enzyme. And what does PFK1 do? It's right here. It's an enzyme that does what to fructose 6-phosphate? It adds a phosphate, so it turns fructose 6-phosphate into what? Fructose 1,6-bisphosphate. So this is the normal reaction for PFK1. It takes fructose 6-phosphate and it converts it to fructose 1,6-bisphosphate. When PFK1 has fructose 2,6-bisphosphate, what happens to the activity of PFK1? It increases. When there's a large concentration of ATP, what happens to the activity of PFK1? It decreases. So do you guys see that with the blue line? Initially it's increasing, but after a certain point, when there's so much ATP, essentially what this means is that there's so much ATP that it's starting to bind allosterically to the enzyme, which deactivates it. Does that make sense? Okay. So the sigmoidal curves are due to cooperativity. What do allosteric activators do to cooperativity? There you go. They decrease cooperativity. That's another important point that I want you to remember. Allosteric activators reduce cooperativity . So allosteric activators reduce cooperativity and they make the enzymes behave more like what? Perfect. All right, so they make them behave like Michaelis-Menten enzymes. All right, and so now we're going to talk about allosteric regulation a little bit more. So a typical example is shown schematically here. The product F of the 5 enzyme pathway acts as an allosteric inhibitor of the first enzyme in the pathway. This is called feedback inhibition. So we have substrate A reacting with enzyme number 1, and once substrate A reacts with enzyme number 1, what do you get? You get product B, and then product B reacts with enzyme number 2, and what does enzyme number 2 do to product B, or substrate B now? It turns it into C. Now let's stop here. What happens if the concentration of C becomes so great that it starts to lower the activity of enzyme 1? So enzyme 1 acts on which substrate? A. And enzyme 1 takes substrate A and it turns it into product B. Enzyme 2 takes this, now it's substrate B, and it turns it into product C, and C is now inhibiting enzyme 1.
So I have a lot of A and few of B and C.
So what's happening is that the accumulation of C is slowing down the activity of enzyme 1, and this is called feedback inhibition. For this example that I drew here, for the one that you see on the screen to the left, what is F doing to enzyme number 1? It's inhibiting it. And the only way that you know that it's inhibiting it is because the slide tells you. Usually there should be a plus or a minus sign. If you ever see a minus sign, that means that it's inhibiting it. And if you ever see a plus sign, that means that it's what? Activating it. So you guys are going to see plus and minus signs on the glycolysis part of your lectures. What is one allosteric activator that I can put here for phosphofructokinase 1 that we've talked about? Yes, fructose 2,6 bisphosphate. So plus F26BP. What does F26BP do to the activity of phosphofructokinase 1? It increases it, so therefore, are we going to turn more fructose 6-phosphate into fructose 1,6 bisphosphate? Or less? More. So that's allosteric activation, and this right here is feedback inhibition whenever the accumulation of something inhibits another one. So an allosteric enzyme like this is called a regulatory enzyme. So all the enzymes which are able to be allosterically regulated are regulatory enzymes. Okay, so number 1, how is insulin activated? It's on your sheet. So is insulin activated by phosphorylation, yes or no? No. What about by the release of an inhibitory complex with another protein? Yes or no? It sounds a little right. So let's put a little squiggly line next to it. What about C? By binding of an allosteric activator. Is that how proinsulin gets turned into insulin? By binding an allosteric activator? No. What about the introduction of a disulfide bond?
No.
No. What about by proteolytic cleavage of the polypeptide backbone? Yeah. So between B and E, I would say E is the best answer. Because it's true. Proinsulin has to be proteolytically cleaved in order for it to be activated. Number 2, allosteric regulation depends on what? The effector binding to the enzyme's active site? Yes or no? Do allosteric effectors bind to the active site?
No.
No, they do not. Good job. What about, do the effectors bind to a site distinct from the active site? That sounds kind of right. So I'm going to put a squiggly line next to it. What about C? Allosteric regulation depends on covalent modification of the enzyme by an effector. Is that true or false? Covalent modification of the enzyme by an effector. Is that allosteric regulation? No. That's covalent modification. What about binding of an effector that is structurally similar to the substrate? No. Why not? Exactly. So if you bound an effector that is structurally similar to the substrate, what type of inhibition do you think that is? Competitive. All right. What about E, interaction of the enzyme with a regulatory protein? Is allosteric regulation dependent on another enzyme reacting with a regulatory protein? Or is allosteric regulation dependent on the substrate of the allosteric effector? So either we have interaction of the enzyme with a regulatory protein, or the concentration of the allosteric effector. Which of those do you think allosteric regulation depends on? A protein or the effector? The effector. Allosteric regulation depends on the effectors. So, do you guys see why E is wrong? If you don't, I can explain it. I can see that some of you were a little confused about that.
Can you explain it?
Yes. So, allosteric regulation. What type of molecules exert allosteric regulation on enzymes? Allosteric effectors. Do we agree with that? So allosteric effectors. And we said that there's two types of allosteric effectors. Allosteric effectors are just molecules. Does that make sense? Remember when we were talking about fructose 2,6-bisphosphate? Is that an enzyme or is that a molecule? That's a molecule. So the example here is fructose 2,6-bisphosphate. So what does fructose 2,6-bisphosphate do to PFK1? What does fructose 2,6-bisphosphate do to PFK1? It increases the activity, so I could put a plus sign there. So is PFK1 being allosterically activated when fructose 2,6-bisphosphate makes it work faster? Yes? Do we agree with that? At any point in time, did I tell you that there's an enzyme which acted on PFK1? No. So what is acting on PFK1? The effector. So allosteric regulation, does it depend on the interaction of the enzyme with a regulatory protein? No, it depends on what? The effector binding to a site distinct from the active site, and that's exactly what fructose 2,6-bisphosphate does. It binds to another site on PFK1 that is not the active site, and it makes it work even faster. Does that make sense? All right. All right, so before we continue, because we're running out of time, let's do this. I'm gonna record the rest on my own time, but we have enough information to answer number 3. All right. So inhibitors of enzymes are important tools for elucidating the pathway steps. For the pathway shown in figure 1, an inhibitor was used to inhibit one of the enzymes in this pathway, and metabolic concentration of each intermediate was measured, which is figure 2. Please use the figures below to answer the following questions. So number 3A, looking at figure 2, which enzyme was inhibited in this pathway? So let's look at the concentration of B. We have a pretty good amount of it. C, we also have a pretty good amount of it. D, same. E, same. What about F? We don't have a lot of F, do we? So which enzyme is responsible for synthesizing product F? E5. So which enzyme was inhibited?
E5.
E5. Does that make sense? Are there any questions about that? So if you're given one of these graphs and you're given like a little metabolic pathway, do you guys feel confident that you're able to answer something like this?
Yes.
Okay. 3B, if intermediate C inhibited enzyme number 1, so let's say C is inhibiting enzyme number 1. So what type of sign should I put next to this? Plus or minus? Minus. So whenever you see a minus, what is that doing? What is that substrate doing to that enzyme? If you see a minus sign,
Feedback inhibition.
that's inhibition. So, thank you for answering it. So if intermediate C is inhibited enzyme 1, what type of inhibition does this present or represent? And what's the answer for 3B?
Feedback inhibition.
Feedback inhibition. All right, 3C, if intermediate C inhibited enzyme number 2, so C is now inhibiting enzyme number 2. What type of inhibition does this represent? Product inhibition, which is a type of feedback inhibition. So the answer therefore is what?
A.
C, all the above. Are there any questions about any of these in 3? Okay, like I said, I'll go over the rest of this on my own and I'll post a video on it, or I'll try to. I should say that. If you guys have any questions, please stick around. And other than that, have a great weekend. Hey everyone, I know that our session just ended, but I feel like there's a few things that I could have said about the glycolysis diagram, which I forgot to mention in class. So I wanted to touch up on that right now. So I didn't actually explain all of this, this diagram, and I feel like I should have done that because I know that these diagrams can be a little confusing, and there's a lot more detail in this other than the arrows and what is written in bold and black. So I want to go in just a little more detail as to why this is so important. So we were talking about allosteric enzymes, and we were saying that allosteric enzymes are different from Michaelis-Menten enzymes. And the reason why they are so different is because these enzymes can be allosterically regulated. And so what that means is that if they can be allosterically regulated, then they have allosteric activators and they have allosteric inhibitors. Okay, so let's go in a little more detail with this glycolysis diagram. And what I want you to do is I want you to ignore everything that is written in blue. And so I only want you to focus on what's on the left-hand side. So if you'll see here, you have glycolysis and you have glucose getting turned into glucose 6-phosphate. And you see this arrow right here, the one to the left of it, probably not because I'm hiding it, but now you see this arrow. Notice how it's not a reversible arrow. We see reversible arrows here and here. Here, let me do that in a different color. We see reversible arrows here and here. So you see there's one going down and one going up. That pretty much means that it's reversible. But if you look over here with Hexokinase, which is the enzyme, you only see a one direction. And the reason for that is because allosterically regulated enzymes control really exergonic steps of a reaction. So these are regulatory enzymes. And they are regulatory enzymes because their reactions are very exergonic. And what this means is that the delta G is less than 0, which means that it's something that is spontaneous. So all of the regulatory enzymes in glycolysis are allosteric enzymes, and what that means is that they can be controlled through activators and inhibitors, which are allosteric. And yeah, So let's go back to the diagram right here on the left. Here you see glucose getting turned into glucose 6-phosphate by the actions of hexokinase. And remember, hexokinase is an allosteric enzyme, so it is a regulatory enzyme. And what that means is that this reaction is delta G less than 0, so that it's very favorable, it's spontaneous. And so then, now that we have glucose 6-phosphate, phosphohexose isomerase takes glucose 6-phosphate and it converts it into fructose 6-phosphate. So here we're going in the down direction. Glucose 6-phosphate gets turned into fructose 6-phosphate, and then phosphofructokinase 1, PFK1 takes fructose 6-phosphate and it converts it into fructose 1,6-bisphosphate. And remember, we said in class that this was a regulatory enzyme, which means that its delta G is less than 0. These are important things for you to keep in mind. If you have a regulatory enzyme, the delta G is going to be less than 0, which means that it's a very favorable step. Versus these reactions right here by phosphohexose isomerase, which are reversible, which means that the delta G is somewhat close to 0, which kind of makes the reaction favorable and non-favorable. This is why it's able to reverse so easily. But the ones where the delta G is less than 0, that only has one single direction to go into. So for that reason, phosphofructose kinase takes fructose 6-phosphate, converts it to fructose 1,6-bisphosphate, and that reaction is not reversible. Don't focus on what's on the on the right. I know that there's an arrow going up, but that's a topic for another review session. Let's just stick with what we know on the left-hand side. Phosphofructokinase 1 takes fructose 6-phosphate, converts it to fructose 1,6-bisphosphate, and that reaction is spontaneous, so therefore it is not reversible. Okay, so because this is an allosteric enzyme, because this is a regulatory enzyme, phosphofructokinase 1 has allosteric activators and allosteric inhibitors. And one allosteric activator that we talked about was fructose 2,6-bisphosphate. And one allosteric inhibitor that we talked about was ATP. So fructose 2,6-bisphosphate is an allosteric activator of phosphofructokinase 1, and it makes phosphofructokinase 1 work a lot faster, and it makes it act a lot faster. So you're going to want to know the allosteric activators and the allosteric inhibitors for all of the regulatory enzymes in glycolysis, which, like we said in class, was hexokinase, phosphofructokinase 1, and pyruvate kinase. But as we proceed in these review sessions, we're going to touch more on these. You just have to know that the regulatory enzymes are allosteric enzymes, and these allosteric enzymes are allosterically regulated through activators and inhibitors. And we're going to go more into that in the next review session. So with all that being said, I hope that you'll have a great weekend, and if you have any questions, please go ahead and reach out to me. Thank you.